Hello, my name is Isabella Mercado, and today I'm going to talk about the P2Y1 receptor in the colonic submucosa of rats and its association with opioid-induced constipation. So first of all, we have to talk about the P2Y1 receptors. They are purinergic receptors that result from the subclassification of the P1 and P2 receptors. They are metabotropic receptors which activate by ATP and are located in the submucosa of the colon. They participate in synaptic processes, activating cellular communication. Now we have to talk about constipation. Constipation is the reduction of bowel movements. When this happens, the stools can become very difficult to pass. Patients may experience the feeling of their colon being blocked and difficulty to defecate. This can be a chronic or can be something occasional. The general objective of the study is to understand how the P2Y1 receptor in the colonic submucosa of rats associates with opioid-induced constipation. Now for the materials and methods, we have the animals. The animals that were used were Sprague-Dudley rats. They were 6 weeks old and weighed between 180 and 200 grams. They were maintained under controlled conditions and were allowed to free access to water and food. Now we have the immunofluorescence staining. This is an immunochemistry technique that uses fluorophores to locate bound antibodies. With this technique, researchers can observe the expression of specific proteins. In this study specifically, the researchers used the technique to observe the expression of the P2Y1 receptor in the colonic submucosa of normal rats. Now we have the reverse transcription quantitative PCR. This technique is used to determine the expression level of specific genes. It uses RNA as the starting material for in vitro nucleic acid amplification. In this study, the PCR was performed to evaluate the relative mRNA expression of MOR and P2Y1 genes. Finally, for materials and methods, we have the Western blotting. It's an antibody-based technique used to detect the presence, quantity and size of certain protein in a sample mixed with other proteins. Proteins are separated through electrophoresis and then the specific protein is identified using antibodies. This was used in this study to evaluate the expression changes of MOR, P2Y1, and ATP beta proteins in the colonic submucosa. So now we have the results, and the first one is the immunofluorescence analysis. So this analysis revealed that a large number of the P2Y1 positive nerve cells were aggregated in the colonic submucosal plexus of rats to form ganglia. So this is what the first image is showing us. The second image is showing us the P2Y1 positive marker located in the cell bodies. That is what the arrows are showing us. The next one, C, is showing us the same P2Y1 staining, and D is showing us the NEUN staining. Finally, image E is showing us the colocalization of the P2Y1 and NEUN cells. On the other hand, the results of the ATPB, the P2Y1, and the CB proteins were significantly decreased compared with the normal saline group and the normal control group, and this is what these are showing us. So this one is the P2Y1, and as you can see, it is decreased. This one is the ATP, it's also decreased, and this one is the CB, which of course is also decreased. So next we have the Western blotting results. So consistent with the reverse transcription quantitative PCR results, the Western blotting analysis revealed first of all that the levels of MOR protein, that is this one, and is the opioid receptor, was increased in the submucosal layer of opioid-induced rats, and that's exactly what this is showing us. As you can see, it is increased in comparison to the other groups. For the discussion, we're going to take some statements made by some authors and see if they go according to what the study said. So first, we have ATP regulates the functional activity of the colon by activating receptors such as P2Y1. This in fact goes according to the study, so the authors and the study that was made agree in this one. Next, we have gastrointestinal induced constipation is associated with abnormal colorectal nerve signaling. This is also stated very clear in the study, so we can say that this statement and the study agree too. For the final statement, we have ATP is an endogenous agonist with a strong affinity for the P2Y1s that activates these receptors and causes relaxation of the colonic smooth muscle, thereby relieving the opioid-induced constipation. And this is very clear in the article. This is very discussed in the article, so this statement also agrees with what the study says. So finally, we have the conclusions. The first one is that the use of molecular biology techniques like the immunofluorescence, PCR, and Western blotting that were used in this study are very important to understand the basis of the causes of many pathologies. The second one is that thanks to all the information that is provided from the molecular biology, treatments for diseases can be developed in a more specific and direct way, focusing on the root of the cause. Thank you.